Hello, thanks again for sticking around. I've really enjoyed the first few talks here. I hope that we continue to have these really interesting talks here. I'm going to talk about something that all of us kind of know intuitively, but I think it's important that it gets said and spelled out directly. And that's that if we want to really apply this technology in a way that's beneficial and cost effective, we need to understand the mechanisms that are driving the effects that we observe. And that's going to involve, so the central thesis of my talk here is that we need to iterate between the lab, the field, and the greenhouse, and that each of these different experimental settings has a place, and that we need to keep this circle of information going between these three experimental scenarios. So I started with the gimmick slide. This is what happens when you search biochar in the news. We see the hype of that our community has created. And then I wanted to put this slide up. This is one of my favorite slides because it really illustrates the complexity of our field results. This is from a field trial in Zambia run by Corneliuson et al. And published in 2013. And they saw yield results that if they're averaged, they match lots of the data that many people observe for yield results, plus or minus about 10% increase in yield. But they see it occurring as a 300% increase at one site and no change in the other four. And so our field results are complex. In the case of this trial, this is a neat experiment because they did do this iteration between the field, the lab, and the greenhouse. They went back to the lab after getting their field results and looked at the properties that changed between these soils. They saw an increase in cation exchange capacity in all 5 soils after biochar addition. They saw an increase in plant available water in 3 of them and a decrease in aluminum toxicity in 2 of them. But interestingly, the one that saw the very significant improvement saw an improvement in all 3 of these properties, and the result is clearly nonlinear. And so it's this nonlinearity that I want to get at today in understanding our mechanisms. So again, just to say this explicitly, we can use lab experiments to really exactly constrain the mechanisms that could be happening. But we all know that all kinds of things can happen in the lab. Things that happen in the lab are not necessarily going to happen in the greenhouse or in the field. We can use greenhouse trials to conduct highly replicated experiments in soil. And so the wonderful thing about greenhouse trials is that you can make N very large, far larger than you can do in a field trial. We can have Ns of 30 to 50 in a greenhouse trial. And this allows us to test the statistical significance of many of the mechanisms that show up in our lab trials. And then our field experiments, when they're informed by greenhouse trials, allow us to take to fully through proof of concept what could be happening. We really need to get at these mechanisms because there's the potential for nonlinearity and non-additive effects. And that's what I'm going to focus on for the rest of the talk. So there are 3 areas that, me personally, I think we really need to know more about mechanisms. We need to know more about biochar's interactions with water, microbes, and we need to understand its sorption of environmental contaminants. And we've seen great talks already today about this. In terms of water, I just want to say that, and again, this is going to be obvious for lots of folks here, biochar's performance hydrologically is a 3-way interaction between the water, the soil, and the char, so it's impossible to predict the hydrologic behavior of a char by characterizing the char itself. It has to be characterized in the soil context that it's working in because the soil infills the pores between the biochar particles. So if you imagine biochar as needle-like structures, the space between those needle structures is filled with soils differentially depending on the kind of soil you're working with, and that has big effects on the hydrologic properties that are finally observed. The microbial systems, I think, are the places where we really have potential for surprises, and that's what I'm going to focus on today. So I'm going to talk about 2 nonlinearity vignettes that are research that's come out of the Rice Biochar group. So one of the hypotheses that gets kind of bandied about is, okay, so mycorrhizae are good for plants. We know that by improving the mycorrhizal infection rate in plants, then you get bigger plants, you get healthier plants, sometimes you get improved crop yield, and biochar can also improve plant growth. So maybe it's double good if you put both of them in, right? Okay, so it turns out it's not double good. In fact, in a greenhouse experiment that a really awesome undergrad in my group did about 2 years ago, he looked at the above ground dry biomass yield for sorghum grown at about 18 tons of biochar per hectare. And what he showed is that in a triple treatment with biochar, mycorrhizae, and nitrogen, he almost halved the above ground biomass yield. So in this case, there's a nonlinear, it's not only not additive, the combination of biochar and mycorrhizae, but the two of them together are acting to reduce the yield of above ground biomass. And in this case, we could see that this was occurring because the mycorrhizal root colonization was vastly accelerated by the presence of biochar. So in this scenario, the biochar was causing very rapid root colonization by mycorrhizae, so rapid that the mycorrhizae were drawing nutrients down from the plant at a rate that the plant could not compensate for, which resulted in a net decrease in above ground biomass. So this is a scenario where a microorganism, which is typically a symbiont, has become a pathogen because biochar has been added to the system. Now again, this is a 4-week greenhouse trial. It's possible that in the initial 4 weeks in the greenhouse, you see this dip in above ground biomass. Had this been an 8-week or 16-week trial, we might have seen a rebound in a different direction. But the statistics are pretty clear for these first 4 weeks. Okay, so what's going on here? Why is biochar acting to change the relationship between a plant symbiont and the plant roots? And we followed up on this not with the plant mycorrhizae signaling system, because that's incredibly complex. That's an interaction between a yeast and a plant, but instead we moved to a simpler system. We moved to gram-negative bacteria to try to understand the effects of biochar and the ability of gram-negatives to communicate. And we worked with a cell-cell signaling compound called, class of compounds called acylhomoserine lactones, which I was really excited to see the previous talk because they're relevant to those nitrogen cycle genes that you detected. They're controllers and sometimes for those genes. So what we saw is that some biochars are very effective at clipping the phone lines in the soils, making it impossible for microbes to communicate amongst themselves and plan their behavior. So what you see here on the x-axis is the surface area of biochar, and on the y-axis is a metric of how effectively biochar is cutting the phone lines in the soils. And I don't want to talking about the method here is beyond the scope of this talk, but I'm glad to talk later about how we made these measurements. So part of the what I wanted to say here is part of the nonlinearity that we were seeing is likely due to biochar sorption of the communication compounds that plants and mycorrhizae use to communicate among themselves and coordinate their symbiosis. In a second, I wanted to give you another nonlinearity vignette, also involving mycorrhizae. So different greenhouse experiment, but still biochar, mycorrhizae, and sorghum. And again, this is another one of those additivity questions. So biochar is good for drought performance and mycorrhizae is good for drought performance. Do we get double good if we add them both together? And again, the result is no. This is also not a system that's either linear or additive. So first of all, the first result that we saw in drought performance, and this is again a sorghum plants grown for about 4 weeks at about 18 tons per hectare of biochar, 450 mesquite biochar. And we don't see this additive benefit. In fact, we don't even see any benefit from biochar in terms of drought. We do see a benefit for mycorrhizae in terms of drought, but not for biochar, and no additive benefit for the two of them. We did, however, see other effects in the system. Because we were working in a greenhouse experiment, we were able to carbon balance the pots. So we looked at the total amount of carbon that was put into the pots in the soil, plus the total amount of carbon put into the pots in the biochar, and then measured the total amount of carbon in the pots at the end of the experiment. So this is a way to get at the priming effect of biochar. And so that red dotted line that you see on the screen will be zero priming. So the red dotted line is what you'd expect if there was no net accumulation of carbon due to a positive priming effect, or no net loss of carbon due to a negative priming effect. And so what we saw was that there was positive priming when we had, under drought conditions, when we had biochar alone. We had positive priming with biochar, with mycorrhizae alone, and especially positive effects with biochar alone. And then when the mycorrhizae and biochar were added together, the priming effect went away. So we're seeing an interaction between the biochar and mycorrhizae, reducing the positive priming effect in the system. So what's going on there? The other thing the intrepid postdocs who did this experiment measured was they measured the aromaticity of the dissolved organic carbon released from the pots. And what they found was that adding biochar to the mycorrhizae treated pots reduced the overall aromaticity of the dissolved organic carbon that was being released. So what we think we're seeing here is that mycorrhizal enzymes are oxidizing the surface of the biochar. So in part, in 4 weeks, it seems like kind of not a positive story because we're seeing this loss of the priming, but maybe it's not a negative story. Maybe it's a story about how mycorrhizae develop that patina on the surface of the biochar that we need to provide these long-term cation exchange capacity sorption benefits. So maybe we're seeing these very early initial stages in the development of the patina. Maybe we're capturing that in this greenhouse, in these greenhouse experiments. So finally, I want to end by saying we really have to get at these mechanisms so that we can make meaningful predictions for where biochar can be used successfully. And we get at these mechanisms through iterative experiments. We do experiments in the lab to get at what might be possible. We reproduce those in the greenhouse at highly replicated conditions to get at statistical significance, and then that allows us to use our field experiments most effectively.